We have been studying chapter number 5, the fundamental unit of life. This is the first chapter of your biology section, okay. And in the last two period we have described the concept of the cell and what is the cell and what is the essentiality of the cell and how the cell functionalizes and why the cell is said to be functional and structural unit, okay. So, you just may see here on the screen yours, okay. This is the what the cell living being is made up of. We have explained already this what the cell living being is made up of right now. So, already we have discussed this about this, and the activity you may see this sketches. These are the compartment like structure you would see here. This first time would have been seen by the Robert Hooke. Robert Hooke observed these types of compartments and the this types of chambers under the slice of in the slice of cork, right, under the microscope right now. So, and this leaded to the discovery of cell, okay. You may see here before this picture is the microscope, and this microscope is used to see the very microorganisms, even the cellular structure is explored under the microscope right now. Microscope was the great discovery of the physics that helped biologists a lot, and after the 19th century's biology grown a 9 days wonder right now. So, simply it is next is what are these structures we have described inside the cell. What the Robert Hooke had seen here, Robert Hooke have seen everything else of the cells but not seen by the Robert Hooke. Robert Hooke have seen the cytoplasmic and dense part of the nucleus he had observed here, and the some organelles he had observed. Not all the organelles were observed by the Robert Hooke first time under the microscope. Is that clear? And we also have discussed about different types of cell. The meaning, the objective of this study or the learning by the student is why we are taught actually the cell concept here, because if we will see the different organism around you, even we could see ourselves. Our body is made up of the different type cells, and all animals body made up of the different types of cells, and the plants is also a living being. These are also made up of the cells here. So, how the functions takes place inside the body, how the organisms actually process its life process. So, the life process like that it's digestions, absorptions and growth and development, reproductions and the fusions, different types of the population you would see here. The population of the tree also grow and the populations of organisms also grow. How this happens here? This happens under certain processes called the life processes right now, and even the organisms take births, and after some years a life is spanning it gets die. So, these all happens due to the cellular process, okay. And how the cellular process takes place, we first have to know the cell structure. So, you may see on your screen the different types of cells are there. What the first type of cell is that here? The first type of cells is a smooth muscle cell is, or this is the human muscle cells you may see, okay. The next what this second type of cell is there, this second cell blood cells of human, and nerve cell you may see the nerve cell is also appearing this like, and how the nerve cells is made up of the structure of the nerve cell if you will see. So, this type of nerve cells is there. We will draw the structure of nerve cell. Simply you can draw the nerve cell, okay, right here, that is this. So, this is the cell body is said to be cytone. Is that clear? Cytone, this is cytone, it is clear. Sorry, cytone, okay, and here you will find a nucleus here, okay, and some cytoplasm you will find a mitochondria and structure is also there, but in each types of cell certain organelles are not present here, okay, and this boundary is further to close here. This portions of the cell is called something else, right. So, this is a typical cell is here, okay, you may see let this is, okay. Further the structure will divide into that. This is a typical cell is before you. This is called neuron or the nerve cells of human or the nerve cell of any organisms. Is that clear? So, what is the structure of the cells and what the organelles, what type of organelles the cell is having. So, we can say life span, we can say functioning, we can understand the process, because the cellular means cellular divisions and the cellular metabolic process and cellular biochemical process actually predict the life of cells, even it predicts the functioning of tissues, that the tissue means group of cells together. If the certain cells are doing the same work, then the group of the tissue is said to be they are said to be a tissue here. In the next chapter we will study here. You may see this next type cell is bone cell is also there here, the bone cell, and here the fat cell also you may see here, the fat cell is also before this is a fat cell right now, and the sperm cell also you may see. So, the questions is rising here, which is the largest cell, which is the smallest cell. So, longest cell is the nerve cell here, longest cell is called the nerve cell, and the largest cell is what here. Have you seen an ostrich egg? Obviously no, because this is found in certain region of the earth. So, we could not see this, we cannot see easily the ostrich, but an ostrich egg, a bigger one here, right. This is a single cell, we can understand here. So, this is the biggest cell of the world, and the longest cell is the nerve cell. Sometimes PP, smallest cell we will talk about. So, what is the smallest cell? So, generally it is spoken as a ppm. So, what is the ppm now? It is a pplo, this is called. What the pplo? What do you understand by the pplo right now? So, pplo is called the certain cells, it means plasmic type structures are found inside this. This is called mnemonic plasmic organisms, look like organisms are there right now. Mnemonic and what is that, what the first is P, mnemonic plasmic right now, and L for what here, like, O for organism, means certain organisms are the bacteria here. Obviously, the bacteria could be, I mean certain definitions are different for pplo right now. So, but the pplo is to be can be written here the smallest cell, but remember you please this, the longest is nerve cell, and blood cell is not smallest here, and here the largest cell is the sperm is also here, you may see this is the smallest appearing here, and ovum, this is ovum, this is the female cell, it is right here. Now, in the female cell it is female cell, this is called male cell, male, and this is called the female cell, clear. So, when they fuse together, then they form zygote, and this zygote further develops into the new young one right now. This we made up of and every child is made up of sperms and the ova also, is that clear? They fuse together to form the organisms new right now. So, obviously, if anyone wants to know how he born, how he take birth on the earth, so obviously we need to know the functionings of the cell right now. So, different types of cell you may see on the screen right now. Now, next is what that we have started the what the cell is made up of, what is the structure and organization of the cell, means what inside the cell we have discussed in the last two period. Plasma membrane also we study about that, what is the functioning of the plasma membrane we have discussed right now, and the process of taking this, like that a cell is before you right now, there is a cell here, okay, a cell I have drawn here, clear. So, obviously, cell will intake the molecules, the small molecules are there, these molecule how will enter into this. If you will see an amoeba, how does nutrition takes place in amoeba. If you will see amoeba how taking food right now, this is like an amoeba I have drawn here, this, okay, an amoeba I am just drawing here, this is an amoeba right, and it's intaking of the food, this is the food is taken by amoeba obviously. So, amoeba is not having any mouth, how it will take here. So, its skin, this is, we can say this is skin actually take the size of mouth here, it means in the next moment, in the next moment the amoeba will make this type of a structure right now, this type of structure the amoeba will make here right now, and the food vacuole will reach up to here, means this finger like projections is being formed here, this is called pseudopodia, it is the pseudopodia right now, it is a pseudopodia. So, how the process is taking place here, and after some time this food prey will be inside the and after that next step the amoeba will develop in this manner here right now, it this types here, the food go inside this and a layer will develop around it. This instantaneous structure is said to be food vacuole, this is food vacuole, okay, is a vacuole, food vacuole, clear guys. So, simply and after the digestion this food, the undigested part is thrown out here from by the body of the process phagocytosis, phagous body, means the undigested part is thrown from the body, is said to be phagous. So, this is the cellular process are here this. Okay. So, what I am telling you this, so you just may see here this how the cell perform its functioning. So, you see this is the outer layer, I am just talking about this outer layer. This outer layer is said to be its membrane, and how the food and the particles enter into this by the process of diffusion and osmosis. In the last period, I explained the process of diffusion and osmosis also in the detail. Right now, osmosis, the movement of the molecules of solvent from the region of high concentration to low concentration via membrane, via membrane, yes, a selectively permeable membrane SPM. This is called the process of osmosis. Is that clear, guys? Understood, everybody? Now, what we are going to discuss here, have looked the next also. So, what is the if the surrounding medium cell has the higher what we will speak here, higher water concentration, then the medium will dilute, gain osmosis. The process of osmosis we have explained here. Okay. Different phenomena also we have seen in our practical life. Is that clear? Now, next is an activity of the osmosis have been shown here. You can also this an activity, osmosis with an egg, osmosis with an egg, osmosis with an egg. Remove the cell of an egg by dissolving into the dilute chloric. If we will take here dilute chloric acid, means we will take a beaker here, and that beaker will put the egg. Is that clear? The egg is to be put into that. We will take here acid will take here this. Okay, we have taken acid into that here. Dilute acid we have taken here, and we will take an egg. Is that clear? An egg we have taken inside this. We will place here an egg. After some times, the bubble will come out this. Okay, the bubble will come out this obviously, and this all the shell will dissolve. The shell will dissolve. So, how the egg will look like? The egg will look like in this manner here. We can say, yes, a membranous structure will found out in this. This thin structure will here there. Okay, it means simply we can just understand here this is an egg, right now, and after this egg will remove its layer, right, and this egg is now put, what is it saying here, calcium carbonate is saying here outer skin, and further the egg, what just I am talking to. Okay, you may see this egg into the by the process of osmosis is also there. Place what's going on here for 5 minutes. Water puts out of the eggs into the cell salts. What is it saying here? Place a similar de-shelled egg in concentrate salt solution and observe. What is to observe here? You may see this is place a similar de-shelled egg in concentrate salt solution and observe for the 5 minutes. We will observe that water passes out of the egg solution into the salt solution, because the salt solution is more concentration. It is very simple here, means if we will place here an egg, okay, an egg we are just taking here, and this egg is now put into where is now the egg is to be put into in salt solution. We will take again a beaker, we will take here another beaker, okay, another beaker will take here this. Okay, in that beaker what will happen here? The egg is put here this. After some time we will observe that this egg gets shrink means this type structure it will have. So, we have speak that we just the water is lost, means egg has lost the water, means water molecule comes out from the egg. Why this happens? This actually happens, you may see this the cells of the water is here, the cells of waters are here, this is the cells of water right now. So, if we will see a microscopical view, so we will see membranous structure will there, and inside the membrane structure this type of structure at that day I told you right now. So, there is the gap between this gap is said to be intercellular space, and water coming out from this intercellular space. Okay. So, this is the movement of the molecules from the region of the high concentration to the low concentration is said to be the process of osmosis. Okay. Well, understood you this method here, this activity. Now, what next is now you are very clear about the definition of osmosis. Now, the activity second is also put dry resins or apricot. Apricots, do you know? Apricot, kismis, right now. Dry resins, water and leave them for a time. Even you can leave them, you can just put them into the night and just see them at the next day morning. Solutions of sugar salt, you will observe that following. Each gains water and will place into the water again. However, when the place into the water concentrate solutions loses water and consequently shrinks. What does this happen here? And everybody know here this, put dry raisins or apricot in plain water and leave them for some times, then place them into concentrated solution of the salt. You will observe the following. What you will do? Each gains water and swell. Means simply you will take a petri dish, will take here dish here, is that clear? It's made out of glass here, or in that you will take water, you will take here water in that, and if you will place here you will put some raisins, the raisins you will put here, this raisins you will put there. The next morning if you will see them, then these will get swell up, well healthy they will appear at this, swollen raisins will appear at this. Why this happens here? So, this happens, swollen raisins, swollen, clear, swollen raisins, swollen raisins. Swollen raisins. Okay. What happened to pen? Okay, raisins. So, this happens due to the entry of the water inside the raisins. Okay. So, in activity also how much water they have, so we can also calculate activity summary is there, activity, this is mathematical activity. Suppose we are let x be the mass or weight of, weight we can just understand here, this weight of raisins, clear, and okay, y is the water absorbed by raisins. Okay, they have absorbed water. So, what is the first weight here? So, obviously the total weight of the this is the soaked raisins. Okay, and how much the water they have absorbed, means x have means this is the water absorbed by the raisins, and this is the weight of the raisins here. Okay. So, simply we can understand here what percentage the solution is there, we can understand here what percentage, just think and make a view about that. Is that clear? I will tell you this after some times. You may just read this what is it saying here. Okay, and this activity is to be done by you. Please read this activity for a 1 minute here, please read this. वो क्या अभी कहीं साइड पर काम नहीं चल रहा ना, इसलिए तो उसको बोलो ना, वो ऊंचा आता था उसको बोलो ना। Okay. Yes, have you read this activity? So, sometimes the activity actually does not come in examination. The purpose of activity that you can observe the scientific phenomena at surrounding you, right, at surround you, right. So, unicellular fresh water organisms, it is simply you would have seen that some waters living in some organisms living in water, and how do they breathe in, how they oxygen they take inside their body. So, they take by the process of simply we can say the molecules goes inside the body and they has to be utilized. So, the concentration become less and again some molecule comes here, right. So, this is the basic principle of the biology right now, means process is to be grown right now, the process must go here and the molecules are to be utilized, and after the utilization of the molecule what will happen, the new molecules will again come here, and if the concentration is to be varied, means pressure is to be made here different between the surrounding points and the inside point. So, molecule will continuously go, just like simple it is. If you eat food, the concentration of the food inside your body, is that clear? Your body, inside your body, is that clear? Inside your body, the less concentration is there. Okay. If the food goes inside your body, your food is utilized, it goes to in your blood stream, and the food just lakes inside in the your small intestine. Again, you just feel hunger, you eat food, and the process go continuously. Is that clear or not? Right? Okay. So, it is very simple. It is further these questions that I gave you. How do oxygen, how do substance like carbon dioxide and water move out of the cell? By the process of diffusion. Is that simply we will write down the diffusions. You will explain the process of diffusions here. Clear? Second question, why the plasma membrane called selectively? Because it allows certain sized particles to go pass through and pass through the membrane. Okay. Now, second, cell wall also explained you. What is cell wall? The plasma membrane have the another rigid outer covering called the cell wall, is made up of cellulose. Now, today we are to discuss here the nucleus. What do you understand by the nucleus? So, you may see what is nucleus. Okay, I am telling this nucleus here. Nucleus is the central part of the cell. What the central part here? What is this? We can say master of the cell. This is okay. You may see there is nucleus. Yes, nucleus. What is nucleus? Robert Brown in 1831, the nucleus has been discovered by Robert Brown, and the nucleus, this is the center part of the nucleus. If you want to draw the nucleus, the nucleus is before you, this is nucleus, clear. If you want to draw the nucleus, the simple way to draw the nucleus is here. Okay. You will draw a rough sketch of the cell, simply a rough sketch of the cell you have drawn here, and make a dotted structure inside this, very dense it is. Okay. This is a nucleus right now, and that it is surrounded by what we can say this is a nucleus, and if you will see the nucleus, is membrane of the nucleus is also found here. Okay. This is the membrane of nucleus, the membrane of nucleus is there. Okay. And if you will see microscopical view, we will see the microscopical view, then how the nucleus will found here. This inside that this coiled structure, we speak here, this the coiled structure will found here. This coiled structure means this type you may see here. There is this. Okay. Let this is this the outer layer, you may see this outer layer is there. Okay. Have a look, this is the outer layer, this outer layer, this is the outer layer. What this is called means is called the plasma membrane. This membranous structure is found here. Inside this structure is there. Okay. Koki. Like this structure is said to be okay. right now. This is plasma. The gaps you may see, what these gaps are called? Holes. This simply are called the pore. They are called pores. They are called pore, and they are nucleus pore. So nucleus. Nucleus pore. It's nucleus pore. Inside the nucleus, you will see a dense part. This is called the nucleus. This is the nucleus you will find out here. And inside that, a coiled structure will found here. This coiled structure you may just understand here. And the coiled structure you will find here. What this coiled structure is said to be? This coiled structure is said to be chromosome. Chromosome. Right now. So this is well defined. Means if you want to draw the nucleus, how can you draw? These are the wall of we can say made up of the nuclear proteins. They are made up of, right now, and these are the pores here. Okay. So three things, and liquid, certain liquid is also filled inside the cell. This liquid is what place? This liquid is filled inside the cell. What this liquid is called? Nucleoplasm. This is called the nucleoplasm inside that. Okay, the nucleoplasm. So throughout the cells, the nucleoplasm will be there, and all they are just merged into this. Okay. So if you just going to draw the nucleus, then how will you draw the nucleus here? Very simple it is. Okay, is the structure of nucleus is before you. So what is the working of the nucleus? The working of the nucleus has been discovered by Robert Brown, and in nucleus is the largest cell structure. Cell structure largest, why it is largest cell structure. Okay, it is a spherical oval prominent structure located. Prominent means it's not necessary here. There we change, it could be also there. Okay. So what next is prominent structure usually located center of the cell. Nucleus, following points are there. These following points are there: chromatin, nucleus, envelope, pores, and the nucleus. This is called nucleus. So what is the working of each part is there? The working of each part you can understand here. This, and before you this. Nucleoplasm. What is the working of the nucleoplasm? It is homogeneous. Nucleoplasm working. It is homogeneous and granular dense present inside the nucleus, which chromatins and nucleoli are suspended here. Chromatin materials, it consists of the long coiled network structure. You may say we have drawn chromatins. These are the chromatin we have drawn. Clear? This is called the chromatins. And nucleolus, what is nucleus? This is called the nucleolus. Clear? So what the nucleus? It is a we can see here this. It is a more or less round shape structure inside this. What this contains nucleus? RNA and DNA are consistent. RNA, ribonucleic acids and proteins. RNA helpful protein synthesis. As the RNA storage, this is called. Clear? And what the next is? Well, understood you about the nucleus. So this is all about the nucleus, and everybody can draw the nucleus in this manner. Right, guys? See the next, please.
भगवान श्री कृष्ण ने माँ आधुरा को छोड़ दिए थे द्वारिका।
Is that clear? Right now? See, and please read this else. कौन सा वाला सामान है? तो वो कहाँ, इसके बाद महावीर वाले पास मिल जाएगा। तो उसके पास मिल नहीं रहा है क्या? ऐसा ना, तो कितने का आएगा? तो मैडम से पैसे ले लेता, फिर 500 हो जाएगा काम। तो एक काम, हाँ तो एक काम करो, मैडम से 600 रुपये ले लो। ठीक है जब
द्वारिका नगरी पहली बार देखा, द्वारिका नगरी डूब रही थी।
वेल्डिंग वाले को बोला है, वेल्डिंग वाले को बोला
कूलर कनेक्ट करवाते हो
is that second? What next further is to be? This is nucleus above right now. So guys, so it's enough for the day, and tomorrow we'll discuss here some more. Is that clear? Thank you, Rachna.